special episode of the Stem Cell Podcast, ISSCR 2025, and we're on day 2. Hey everyone, this is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. Today, we're back with another special episode to discuss highlights from the ISSCR 2025 annual meeting taking place in Hong Kong. If you're at the meeting, make sure to drop by the Stem Cell Podcast booth in the exhibit hall to meet the team. You can also sign up for our delegate recordings if you're there in time. We're doing that tomorrow. Today, we'll be discussing some of the most interesting research we saw presented on day 2 of the meeting. So if you were in another session or weren't able to attend, we've got you covered. We're going to kick things off in just a minute, but before we get there,
did you know that you can generate more than 40 different cell types and organoids with STEMDiff HPSC differentiation kits? Explore the possibilities for reproducible, high-quality HPSC differentiation with easy-to-use serum-free kits at stemcell.com/STEMDiff.
You know, the highlight for us, I'm sure, we said it yesterday, every year, now that we're doing it on the reg, is this live episode, which is just so cool. Different form for us. You know, we're used to being kind of in a safer space, recording and editing these podcasts, but that live show really changes the game. But more than that, it's about engaging with both the guests personally in the same space, but also with the listeners and the crowd that's there for the show. I was very pleased and privileged to have my wife in the audience. And it made such, my wife's not a non-scientist. Love you, Maya. But we don't really talk about science at home. But the great thing about this forum is that she was in the audience and came out of it with so many questions, so much interest. You know, we spent the rest of the night talking about this landmark study, which really does deserve a lot of discussion. And also the insight and inside take from some of the authors of the amazing science. I mean, you know what I'm talking about, I'm sure. But in case you don't, this is the chemically induced pluripotent stem cells that were applied to treat and effectively cure patients, an episode that's going to air officially on June 17th. But it was just so great for me, Arun, to have my wife in the audience, but also to have all of the listeners and a lot of the people in attendance at this meeting there to see that personal take. What'd you think about it?
Yeah, absolutely. A fantastic show. I was really pleased with the turnout. It was around 160 people who were in the audience for that live show. And the dynamic, like you're alluding to, it's unique because typically we're isolated either on different sides of the country, recording interviews and roundups, or like right now, it's just you and me in a isolated room. Of course, things are a little different when there's 160 people in the audience, different dynamic. But that's exactly what we told our guests, Dr. Hong Kui Dang and Dr. Candice Liu, that this is just a conversation that we're having. Don't worry about the people. Don't worry about everybody staring. We're just having a chat about the really incredible science that they've been doing over the last years. First on the development of the CiPSC program and technology by Dr. Dang, and then ultimately the application for differentiation into functional pancreatic cells. And like you said, the effective cure of this individual. I mean, they made an analogy, or they made a reference to how this patient is now able to overdose on ice cream like multiple times a day or something, and their insulin regulation is still there. So that really personalizes the story. I thought that was a really insightful and just funny anecdote from Dr. Dang. So I was just really super happy with the turnout of the show and how it went, and can't wait for next year as well.
Yeah, and then it was on to the concurrence, where I got a bit of I don't want to call it a hot take because this isn't an original thought. Arun and I were in different concurrents in the morning. And we were in the shared concurrent later, which we'll come around to that. Great minds think alike. But I was in the morning concurrent about these adult models of animal regeneration. And I just want to say out the names of everybody, because it was, as we said yesterday, the emphasis being on investigators who hadn't presented before. So these weren't even early career investigators, but they were a lot of new faces, which I love about this meeting. It started with Mesa McCollid, who's talking about zebrafish and spinal cord regeneration. Then there was Ernesto Almada from USC talking about the green anole lizard, which was crazy. I'm going to come back to that. Yi Sun from Shanghai Tech, also talking about zebrafish, in this case, the fin regeneration. Kai Lei from Westlake University, talking about planaria. And then Hui Zhang from Chinese Academy of Science, talking about neonatal apical resection, combining that with human organoids. And then finally, Tatiana Sandoval Guzman talking about the human fingertip regeneration. So we were all over the map in these regenerative models, all the way down from planaria up to the human. But the takeaway that I had here, and I just want to say it out here for thought and for discussion, is that what I really noted, and I came there, and I said this yesterday, I went for the lizard because I had a personal connection in Trinidad chasing these little lizards, but also my wife was in the Caribbean a few months back, and don't tell the Fish and Wildlife Service, but she accidentally smuggled back a brown anal lizard that we took care of, named her Lizzie, ultimately gave her to the pet store, so they got to deal with it now. They're the high maintenance, these anal lizards, probably because of their amazing capacity for muscle generation, which is what Albert talked about. Which, it was a great story about tool building. And all these stories were about how you're applying these novel, kind of all this tool set that we have to go from space here in the lizard with single cell seq and lacking any kind of genetic models to glean some insight. It was really a fascinating story. And I guess that's my point that I want to make about the story. And in these forums, it's about storytelling, telling your scientific story. And you can't tell your story without language. Here on in this podcast, we obviously have a love for communication, love for language, and the amazing privilege that we have as native English speakers, or I should speak for myself, the amazing privilege that I have as a native English speaker, to be able to tell my story in my native language, I think is something that we don't talk about enough. And of course, we're really enriching for a lot of the Chinese talent. We're in China talking about stem cell science. And there's so many stem cell scientists here, but people can't tell their story, their scientific story in their own language. So I just want to highlight that people are at a disadvantage in this, where we talk so much about, equitable and inclusive practice of science. And I think we're doing well. But I think that it's going to be really great moving. And I'm not just hating here, because I can see a near future where scientists are able to tell their story in their native language like they do at the UN. And you have a real-time, AI mediated translation of that. Because frankly, it's harder to convey their message with such a nuanced and to display their charisma and enthusiasm in a way that I really saw with Dr. Almada. He was telling the story about how he was inventing this model system, and I felt that. And I'm sure every one of the researchers here are so pleased to share their story, but they got to do it maybe in a foreign language. And the intelligence that it takes to be able to do that, I'm in awe of. But I just wish we could acknowledge that and maybe accelerate this transition to a real live recording, especially for some speakers for whom it may be a little bit more limiting in terms of the forum that they can address with their science. So that's my piece, not an original insight, but I just wanted to acknowledge it here in this meeting that is so focused on equity. I think that there's something that we're an elephant in the room that we're not talking about enough.
That is an absolutely fantastic point, and those AI technologies are available. You could put in your earbud on your AirPod and actually do that live translation with an app that is available on the App Store. So you're absolutely right. Within the next 5 years, perhaps this is going to become a reality, since those technologies are so accessible. And absolutely agree with you. Shout out to the folks who are presenting not in their native language, because of course, English, fairly or not, is the medium of modern science currently, although that could be changing. Absolutely. It terrifies me, that idea of not presenting in your native. I just can't imagine doing that. So it takes tremendous courage for all the folks, not just the Chinese scientists, but folks from around the world who are communicating in English, whether that's your your native language or not. So I also had really enjoyed the morning sessions. I just am blown away by the quality of the talks today, just across the board, from the plenaries to the concurrent sessions. In the morning, I was focusing more on the disease modeling side of things, since that's pretty much what I do in the lab. And I wanted to learn more about it. So I was at a session called Disease Modeling in Stem Cells and Drug Discovery, from Concept to Application. And the two chairs are, of course, icons in the disease modeling field: Shubing Chen from your neck of the woods in New York, and also Martin Pera at Jackson Labs. Wanted to start things off with Paul Peng from Greenstone Biosciences, who's actually, that's the company from Joe Wu, my PhD mentor. They're developing this concept of a clinical trial in a dish using iPSCs for drug discovery, precision medicine, really building on what the FDA Modernization Act 2.0 has been conveying, that maybe there's a need for to move beyond the animal models for these in vitro toxicology and drug discovery pipelines. And that's exactly what they're trying to do here. They've got a gigantic IPS biobank with 2,500 lines. They're doing toxicology screening and a cell village approach to figure out who is going to perhaps have propensity to develop cardiotoxicity in response to a toxic agent such as doxorubicin. And the nice thing about the cell village approach, where you pool everything together, is you don't have to ask some poor research assistant or whoever to do 2,000 independent IPS cardiomyocyte differentiations. You can just pool everything together in a single dish, do one differentiation, and then do a single cell RNA sequencing analysis to figure out what's going on phenotypically. So that was really great to see. Moving on to Yuting Fu from Westlake University here in China, focused on iPS-derived neuromuscular organoids for disease modeling and drug screening. Really cool, scalable neuromuscular organoid production system that has both the neurons, the skeletal muscle, and you can use this for modeling something that Clive Svensson is very familiar with. That's ALS, since ALS has this dual neuromuscular phenotype. It's a perfect system to study that. Next talk there was Lotta Oikari from Kaima Berghofer, prioritizing drug candidates for Batten disease. And again, another disease model, IPS differentiation into cortical neurons and astrocytes. And the hope is to identify drugs that can, again, alleviate specifically lysosomal dysfunction, which is the hallmark of this Batten disease. Liam Allen Reed from the University of Nottingham focused on multicellular iPSC organoids, alveolar organoids to model pulmonary fibrosis. This is a xeno-free platform for the co-culture of 5 different cell types relevant to pulmonary fibrosis. And as you might expect in fibrosis, TGF-beta signaling was a critical pathway. And then finally, wrapping up with actually a couple more folks, Juliana Moriar Marquez from U of Sao Paulo, differentiating pituitary cells from iPSCs, making functional pituitary cells and organoids for modeling pituitary disorders. Part of this was published already in Cell Stem Cell, but they further refined this protocol and applied it to a Prop1 mutant system. And then the last talk for that session was Benedetta Artigiani. We've actually covered some of her work before from the Princess Maxima Center. These really neat brain, liver model systems from primary tissues to create organoids for studying human disease. They're scalable, they're primary, this is the real thing. They call them fetal brain organoids. That's what they're derived from. It's a fetal tissue that they turn into these self-sustaining organoid system. They already published a Cell and Nature Protocols paper on it, but they're extending some of these studies to, again, a more advanced disease model. And then we'll move on to the plenary 2, which was development across scales from fertilization to tissues in animals. First, leading off with Tao Tan from Kunming University, gave a really nice overview of the 3D modeling and embryo development models that are out there right now. A lot of parallels between monkey and human models for in vitro development, as you might imagine. They examined or presented some unpublished data looking at mouse development ex utero up to the organogenesis point, and a really just nice general overview of the key embryo model systems that are out there. I forget who mentioned this, but someone said what we've said multiple times here on the show, that this is the hottest area of research in stem cell biology. They straight up said that in the plenary session, which I was happy to hear. Of course, the epigenetic signature for a lot of these early embryo models is still not very well established. Still got to figure that out. Moving on to Bo Wang from Stanford. This is a really cool talk on regeneration suppressors and the power of unique model systems, like you're talking about the lizard, the power of the planaria in this particular example as a model system to study regeneration. It's an awesome system when you get full body reset after a significant injury. ERK signaling is really critical to the regenerative capacity in the planaria, and they're able to identify some regenerative pathways using RNAi, blocking them off, and seeing then increased cell proliferation.
Yeah, that Bo Wang story was so captivating to me, and his presentation, the way that he laid it out, I thought was, I mean, artful. Just a conceptual, an idea that he introduced with from all black slide with just the print, the idea of regeneration suppressors, and just as soon as he puts that up there, you're going to get a talk that's going to kind of change the way we think about things. And then just a feast for the eyes with all this planaria imaging and animation that really drives home the power of this, simple animal model in gleaning major insight here. And of course, he left the cliffhanger, inviting the whole crew there to collaborate, which I'm sure there was a lot of people whose mouths were watering on that front. In terms of how these questions, these ideas of regeneration suppressors exist in vertebrates and beyond. So really exciting. And within this talk, the plenary, I thought that it was, again, really well organized, going from Tao Tan talking about these embryo models, as you described, and the advances thereof, going back to the planaria, and then talking, 10,000 foot view here with Misao Fujita, who's from Kyoto, talking about a really out of the box thing. And I remember talking about this in the pre-meeting show that we did with the leadership, that part of the idea was to take kind of these niche talks that you usually see in the sideline and the concurrence, and elevate them to the plenaries where you have the attention of the entire audience. And that was the case here. It was a bit of a hybrid, though. Dr. Fujita was talking about the public's reception of in vitro gametogenesis, but they did a very well designed study survey of the public. In this case, 3,100 roughly citizens, presumably Japanese. And I thought it was something that both confirmed thoughts that I had, but also really gave me a much more granular view of what the perception here, in this case, about many questions related to in vitro gametogenesis. And ostensibly the pretext here for the study was to see whether or not the public was receptive to the idea of loosening the restrictions right now that exist in Japan on generating embryos, of course, from in vitro derived cells, not implanting them or any kind of gestation, but just generating embryos. And the takeaway was they did a little minor education before, and then gave them the survey. And what I learned, one, is that the public, at least the Japanese public, is amenable to education. I mean, these people understood these questions, and I think we're able to give informed opinions. The other thing that was interesting is that there's really differences between culture, just in terms of comfort level of different types of applications for in vitro gametogenesis. I thought it was interesting that Japanese group, kind of their barrier was they weren't that okay with in vitro gametogenesis being applied for same-sex couples or menopausal couples. So there was a kind of a line there. Whereas similar studies have been conducted in the Netherlands and I think Belgium were kind of different in that they were also not receptive to the idea of menopause, but they were open to the idea of same-sex couples, right? And perhaps the history there informing that general opinion, as in the Netherlands and Belgium were the first place where same-sex marriage was approved. But the line being drawn at menopause, I think, was really interesting, because I think the idea here is that we need to provide this option for patients for whom they don't have any other options. I've been outspoken that I'm not okay with it in principle, given the safety profile now, but it was really a great insight to me to get such a granular take on what generally people thought about it. So I was really glad to hear that, and I'm really glad they elevated it to the plenary. And then, of course, there was the McLaren, which I always love the McLaren because it has a focus on IVF. But this year, it was taken to, I think, a new level, arguably. Why don't you tell us about the McLaren?
Yeah, really great talk from Hongmei Wang from the Institute of Zoology in the Chinese Academy of Science. And the title here was, deciphering the mechanisms of primate embryogenesis, fit really nicely with the rest of the talks here. And as always is the case with the Anne McLaren Memorial Lecture, they always start off with a beautiful recap and a tribute to Anne McLaren and the impact of her work, and in particular, the very early work that that was done, and ultimately, of course, led to the development of IVF, which is a medical mainstay. Really great talk, and just a reminder, quote, that gastrulation is indeed the most important event in your life, more important than birth, marriage, or death, and that's a very famous quote. And there's just a nice recap of early primate work, in vitro modeling, opening up that black box beyond day 14, between day 14 and 28. Some really amazing unpublished single cell data was shown looking at post-gastrulation human embryos derived from IVF clinics, and that was really cool to see. Also, development of different human placentation models, such as these endometrioids to model blastoid invading into the human endometrium. So again, very early model of human development. And they had to wrap things up. She had to wrap things up with a reference to studies done in space. And you might wonder why this is relevant beyond the fact that I just love this work and I do this work. This is a question that ultimately will need to be answered. Can human development happen in space? How is it altered in space? Eventually we're going to be spending more time into space. So apparently there's already been a project sent to the Chinese space station where they looked at using these early mouse embryo models, sent them to the space station, and were examining the mechanisms of their continued development in a microgravity setting. So obviously, I thought that was the coolest thing ever.
Gotta worry about them belters, don't we?
We do love The Expanse here. Check it out. Check it out if you haven't already. Yeah. So moving on to probably one of my favorite concurrents of all time. This was in vivo and in vitro organ generation. This was just a feast for the eyes, and just beautiful visuals and beautiful model systems that were being presented here. This was hosted by Jürgen Noblich, of course. He's been on the show. First, guest really needed no introduction. First speaker, this is Hiro Nakauchi, somebody whose work I truly love and am inspired by. He's at Stanford, but also has a cross affiliation with Japan as well. And if we're talking about Hiro Nakauchi, we're talking about chimerism, certainly. And this is a continuation of the chimerism story, which first came out back in 2010, where in Cell, where they show that they're able to generate this rat mouse pancreatic hybrid chimera, I believe. And he was looking a little deeper into it and figure out that there's this idea of host macrophage xenophagocytosis, which is a barrier to interspecies chimera formation. And there's apparently these macrophages that are potentially eating the xeno cells from the host population. And when you modulate and basically prevent the function of those macrophages, you get, as you might expect, by better chimerism. But there's also apparently a secondary function for these macrophages where they're not actually eating the xenocells, or just like cuddling them or hugging them. We've used that phrase before, the cuddling macrophages, right? So there's a lot to this story. There's a lot of elements that need to be improved so that the chimerism mechanisms and the chimerism efficiency can be improved. So there's still a lot of work that has to happen here. They incorporated, of course, the CD47 don't eat me signal, where you get better chimerism because those macrophages are not able to gobble up the xeno tissue. So really great intro there from Dr. Nakauchi. Moving on to someone else from Stanford, Katya Weinach. All these talks were just 10 minutes long. Really great snapshot of what's being done in this field, just across different tissue types. Katya was focusing on iPS thymic epithelial cell formation to recapitulate key functions of the human thymus. They developed a protocol in their lab for making thymic organoids from iPSCs, which was quite a journey in itself, and pretty comparable to the real deal in terms of the single cell transcriptomic analysis. And ultimately, she wants to use them for transplantation applications, putting these iTechs potentially in people who have diseases of the thymus where they're not able to have a thymus or their thymus is degraded. So it's a thymic replacement cell therapy, potentially really cool application there. Moving on to Irene Talon, who did a great job in spite of some technical difficulties from the Max Planck Institute. She developed this self-organized multi-organ bud model from pluripotent stem cells that's able to recapitulate the hepatobiliary organogenesis mechanism. This, again, PSC differentiation into self-organized biliary organoids and really great expression of the key hallmarks of these cells like albumin secretion, other things that they're supposed to make. A really beautiful self-organization, which was, I think, a theme of this concurrent. You put the cells in the right context, in the right niche, and they just form what they're supposed to form in a native architecture or a native-like architecture. So that was really cool to see. Sean Wilson, who I'm sure you're going to talk about this talk, had just an incredible visual representation of how he was able to generate these multi-lineage kidney assembloids with like a polarity to them, with integration between the nephrons, and ultimately creating it almost like a mini kidney. And it's not just like it looks like a mini kidney. It's the visual that's really appealing here. There's a single exiting collecting duct, sort of analogous to what you're getting with the kidney. Really cool. Again, spontaneous organization. Once you combine these different organoid types to the single output with a ureteric epithelium forming in sort of the right way. Ziling Shen, who I've actually heard his talk before he came to Cedars-Sinai and presented some of his work, which is pretty wild. He does a lot of different things. Engineering, again, in the theme of chimera formation, engineering interspecies chimeras with embryonic organoid engraftment. This was an extension of a science paper that he published a few years ago where they developed like a live imaging window of mouse development, just like building engineering a window into the mother's stomach or the uterus area where you can actually visualize live the development of the developing pups. So they use this as a way to visualize the chimerism model. And what they did here was they introduced adult and pluripotent stem cell derived organoids into the system, examined the integration of these organoids into the developing mice, and we're able to modulate that somewhat. And we're able to show that there's like a tropism that happens. So the brain organoids go towards the developing brain, the liver organoids go towards the developing liver. Again, the mechanisms are not well established here, but it's largely an observational study looking through this window. But really interesting tropism that's happening here, where an organoid is going to its ancestral tissue type when it's introduced into the mouse architecture. That's really, really cool. And then wrapping up with, I think, quite a milestone from Shangli Lai at Guangzhou University, xenogenic generation of humanized mesonephros and heart in pig. And the key here, this is again an extension of chimera studies. The key here to improving the efficiency of these pig human chimeras was the induction of these naive custom human pluripotent stem cells in a custom 4CL-NB media, which they've published previously. This enabled a really improved chimera efficiency here in this pig human model. Human cells going into the pig. You have a functional connection with the host tissues. They talked about some of the published work with the mesonephros, but then I believe presented some unpublished work with the cardiac chimerism. And as a cardiac biologist, I was super hyped and super excited by this. Of course, the application is obvious, right? You want an unlimited supply of human hearts or unlimited supply of human kidneys for transplantation purposes. And the thought is you can use the pig as the host to actually grow these human organs. And they were able to show that indeed, using an NKX 2.5 and myosin heavy chain 6 knockout pig, they complemented with this, advanced custom human iPSC source, and they were able to indeed develop an early stage pig human chimera with, get this, a beating heart. Wow, that was an incredible visual to see. I can't believe I saw that. That was apparently the first demonstration of the generation of a developing human kidney heart inside large animals by an interspecies embryo complementation. Of course, these were not carried to term. Duh. I mean, ethical issues and all that. But just the visuals were just unbelievably striking. What do you think, Daylon?
Yeah. Uh, and it's funny because it wasn't like a spectacular image, but the impact, it was a bright field, a little think, think, think of the heart. But the implications of that are profound. And again, a very well designed session. This session was rich, but anchored on at the front end with this chimerism story, which I love. I love the design of that. Two at the end, really, when you think about the embryoid body chimerism from Zhiling Sheng. But the lift that it takes for these chimer models is so tremendous. That's what I was left with. You need so many things to go right, and the efficiency is so low. I mean, the numbers mentioned for that mesonephros study by Langzhu Lai there at the end, it was 1,800 embryos, chimeric embryos, into 13 surrogate pigs over the course of many experiments to get 5 normal fetuses. So this isn't something about, efficiency or scale, obviously, yet, but I think conceptually it's really super rich, and particularly with Nakauchi. It's funny, a lot of the speakers in this session cited him as an inspiration for them even venturing out on this path, because who would have thought it was even possible? But him going next level now with this idea of the cell intrinsic modifiers of efficiency with the macrophage, I think we're really taking this to the next level. But I was there and was really blown away by the organoid assembloid type models which you described. It's just the principle for me, the principles that were gleaned from that, which I feel like is where we are now in terms of improving the efficiency. It comes down to a few things, and one is getting the right starting material, right, with these 4 cell-like cells that are more potent, and maybe augmenting them, but also where do you start? What path do you take? Anterior posteriorizing these early endoderm, or in my case, intermediate mesoderm. I'm really struggling with getting them at the right point along the anterior posterior axis. But then, of course, the assembloid idea, which I think Sean Wilson illustrated so well, is that sometimes you need to take 2 cell points of different origin. They come down different paths, but in development, in developmental physiology, they interact and come back together. There's this kind of heterocrine signaling. I don't know if that's a thing already, but if not, I'm coining it, where you get these 2 cell types coming together in this assembloid context to generate the final organ. And I think that's appreciated more and more, and was really illustrated well by this kind of pseudo-engineering, cellular engineering approach that has been in use for a long while to generate blastoids and similar gastroloids. But here, I think being applied, and more being applied toward more advanced developmental stages. So that is super exciting and really beautiful to see. It's one of those dreamlike moments. We talked to Sean for a minute after his talk, and he was reminiscing about the day, still has it on his whiteboard, the day he first looked into the microscope and saw these organoid structures. And that's kind of those aha moments that we all really dream about.
Yeah, and it's about the people. We've said this before. Science is about the people who do it. It's so great to see everybody here in the flesh, in person, connect some faces, like what you're alluding to the incredible science that's being done. And it's so important for the trainees in particular to get this FaceTime, literally, with the senior folks in the field, because ultimately that leads to opportunities, that leads to jobs. I mean, that's how I got my postdocs and other opportunities, as well as, being able to present at the biggest stem cell meeting of the year. And I was also so fortunate to catch up with some old friends of mine. I had lunch with the Wu clan. Joe Wu and Sean Wu, of course, were my graduate mentors, and I had lunch with 4 or 5 of my closest friends from the Wu labs. That's Mingxia Gu, of course, at UCLA. We've had her on the show. Yan Boykama, Soa Lee, Joe Zhang. Shout out to you guys for catching up and reminiscing for a few minutes with me. But that's what it's all about, right? It's about getting those connections, establishing those friendships, and most importantly, maintaining them over the course of your scientific career. So I hope for those of you out there listening, I hope you're out there networking, making some friends, making new friends as well. And that brings us to the end of our day 2 recap here at ISSCR 2025. Don't forget to follow us on X at STEMCELL Podcast or Blue Sky at STEMCELLpodcast.com to find out what we're up to at the meeting, and visit us at the STEMCELL Podcast booth on the exhibitor floor, where you can win some prizes, and check back here tomorrow for another episode recapping day number 3 of the meeting. See you then.